Today I'm going to be talking about using link prediction in networks to perform drug response prediction. And so the thought behind this is really based on personalized medicine. We're specifically looking at cancer, and we all know cancer is a highly complex disease where patients with the same cancer, even in the same cancer subtype, can have different mutations, different driver genes. And so really their molecular profile can be variable. And so when you use a method where therapy treatment where you get one drug for each cancer type, there's quite a few patients that don't actually benefit from the drug. So the goal then is to identify accurate personalized therapies for each patient, where we use some sort of biomarkers to decide on the drug. And so this is an interesting problem, and it's also very difficult because you have to find which features are best to use from the biomolecular profile, and then how to actually leverage those features in a predictive model to choose the drug. And so how we would approach this was to use cell line mutations, which haven't necessarily been the best predictors as before. Usually gene expression is used, but we try and overcome this by using protein-protein interactions. So immediately we add functional context to the features that we use in our predictive model, and we incorporate this with drug response data from a large cancer cell line data set, and we incorporate it in terms of a large heterogeneous network. And by performing network proximity, namely random walk with restart on these different instances of the network, we can create what we call network profiles for cell lines and drugs. And by comparing these profiles, we can calculate a score for each cell line drug pair. And so that score represents the sensitivity in that case. And so we have for two classification problems where we focus on the classic leave one out cross-validation of the data set that we use to build the network, and then cross-study classification where we bring in another very large cell line study, the CCLE. We see in both cases that we achieve very high accuracy performance by AUC greater than 0.8 in both cases. So that tells us that the model is quite accurate and very robust, and that is able to work on brand new cell lines as well. And you can access this this method at this website at the bottom of the screen. And I'll show you a brief description about the method, link to the publication, and also a user manual, where the user manual tells you how to perform these cross-validation and classification as before, which is discussed in the paper. And then we also have a user case method where you can provide new samples that you may be interested in your lab, and we can predict the sensitivity for those samples to drugs that are in our network. And so this can be used for, as I said, a cell line of interest. It can even be used for drug resistance cases. And all you need is an input mutation file that looks like this numeric matrix where the columns contain the mutations of the cell line, and the mutations are in interest ID. And we can output a ranked list of all the GDSC drugs, which are 138 drugs, where they're ranked by a relative score from most effective to least effective. And so this can be used in many different cases for different types of research that you want to do with cancer drug cell line sensitivity. And so just want to thank you and come by my poster if you have any questions. Any questions now? Right, yes. That result you can see at the poster. Right, yeah, there's 11 drugs in the GDSC and the CCLE that are shared. And there are 235 cell lines in the CCLE that are not in the GDSC. So we can treat those 235 cell lines as brand new cell lines for prediction. And that's really what this AUC value is at the bottom here for CCLE. Those are the predictions for all the cell line drug pairs that were new but represented in the CCLE. And so we, as I mentioned, we have good performance on those as well.